Good day, this is the Professor, CBMD PhD, and today is Monday, September 4th, 2023. It is 3:35 and 31 seconds, AM Eastern Standard Time, and today's research, CAR T-cell therapy. So let's hop to it here. Expanding the impact of CAR T-cell therapy, an immunotherapy strategy against all blood cancers. Date August 31st. 2023. Source: University of Pennsylvania. A broad new strategy could hold hope for treating virtually all blood cancers with CAR T cell therapy, which is currently approved for 5 subtypes of blood cancer. Scientists have demonstrated the potential efficacy of this approach in preclinical study. The scientific investigators and microbiologists used engineered CAR T cells to target CD45, a surface marker found on nearly all blood cells, including nearly all blood cancers. And because CD45 is found on healthy blood cells too, the research team used CRISPR-based editing to develop a method called epitope editing to overcome the challenges of an anti-CD45 strategy, which would otherwise result in low blood counts with potentially life-threatening side effects. The early results represent a proof of concept for epitope editing, which involves changing a small piece of the target CD45 molecule just enough so that the CAR T cells don't recognize it, but it can still function normally within the blood immune system. Up to this point, they haven't had the tools to create a targeted cell therapy approach that could work across all different forms of blood and bone marrow cancers. They are excited to create a new solution that can solve a major issue in immunotherapy, which is the inability to target surface markers that are found on both cancer cells and healthy cells. Each of the currently available cell-based immunotherapies for blood cancer is designed to work against a narrow range of malignancies based on their target antigens. For example, the first CAR T cell therapy. Researchers target the CD19 protein marker on B cells to treat B-cell lymphomas and leukemias. 4 of the 6 CAR T-cell therapies currently approved to treat blood cancers target CD19. The other 2 target the BCMA protein marker to treat multiple myeloma, while CAR T-cell therapy has been remarkably successful. Researchers across the world are working to make it even more effective for more patients. One drawback of the current approach for CAR T cell therapy is that each therapy must be developed individually based on the targets for that cancer type. And because CD45 is found on nearly all blood cells, it is usually highly expressed on blood cancer cells. A treatment that wipes out all CD45 bearing cells would leave patients without any blood cells, including red blood cells, platelets, plasma, and even the marrow-based stem cells that generate new blood cells. And furthermore, since T cells are blood cells and normally express CD45, CAR T cells targeting CD45 effectively would kill each other before they could be infused into patients. The team built on previous work to overcome this challenge using CRISPR-based editing to develop a new strategy called epitope editing. This involves a genetic modification of both the CAR T cells and blood stem cells to alter a small piece of the CD45 structure or epitope where the CAR T cells bind to the CD45 molecule. The altered version of CD45 still works, but differs enough from normal CD45 that the anti-CD45 CAR T cells do not recognize and attack it. It is essentially a blood stem cell transplant paired with CAR T cell therapy. The idea is that when the engineered cells are infused, the CAR T cells kill the cancer cells that bear normal CD45 but don't kill each other or the newly engineered blood stem cells. This allows the engineered blood stem cells to begin making new blood cells. And because the strategy results in replacing the stem cells that create new blood cells, it also has potential use as a milder form of chemotherapy conditioning, which is given the patients before a bone marrow transplant to suppress the immune system. The researchers tested the strategy in an extensive set of experiments in cell culture and mice models. They showed that the new approach not only keeps anti-CD45 CAR T cells from attacking each other or stem cells, but also enables swift destruction of blood cell catchers. In one test, the anti-CD45 CAR T cells eliminated leukemia cells within 3 weeks of infusion and were still present and capable of killing leukemia cells more than 2 months later. And further toxicology studies and additional modeling studies are currently underway in preparation for an investigational new drug application before it can move into phase 1 clinical trials. And this is a professor of microbiology, bioinformatics, pathophysiology, and leader of research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.